Hi, my name is Michael Lott, and in this short video presentation, I'll be going over a significant breakthrough in our understanding of cancer. But first, what is my inspiration for making this video? Recently, my grandmother passed away after a 4-year battle with liver cancer. Her death inspired me not only to learn about the intricate science that we know today, but also to learn about further breakthroughs that can help society in the future. Let's begin. Cancer is currently a significant problem in our society today. 1 out of every 4 deaths in the United States is due to cancer. So if we took a group of 100 deaths, 25 of these deaths is due to cancer. Out of these 25 deaths, 90% of them are due to metastasis. What is metastasis, you might ask? Metastasis is simply the spread of a tumor from one organ to another organ. So let's say I have a primary tumor that developed in my lung cells. If these lung cells move to my brain cells, or to my liver cells, then metastasis occurs. So what is metastasis responsible for? Metastasis is responsible for the upgrading of a tumor. So as you move from stage 1 to a stage 2 tumor, a stage 2 tumor to a stage 3 tumor, or a stage 3 tumor to a stage 4 tumor, what happens in between is that metastasis occurs. Now how does metastasis occur? First, the tumor cell must go from the primary tumor and invade the bloodstream. This process is called intravasation. After this, once the tumor cell is in the bloodstream, it must travel through the bloodstream to a distant organ site. After it reaches this distant organ site, it must invade the membrane and proliferate. This process is called extravasation. So after extravasation occurs, the tumor has successfully established a secondary tumor and metastasis is complete. So what are present limitations in our knowledge? First of all, as I just demonstrated, we know how metastasis occurs, but we don't know why it occurs. Now, this has severely hindered the effectiveness of clinical treatment, such as chemotherapy and radiotherapy. What do chemotherapy and radiotherapy have in common? Both of them target the primary tumor, but both ignore the metastatic process. We don't do this on purpose, it's just that we simply don't know how to target the metastatic process because we don't know why it occurs. So my breakthrough really aimed to solve this, and my breakthrough is the process known as Epithelial-Mesenchymal Transition, also for short EMT. Now what is EMT? The Epithelial-Mesenchymal Transition is a biological process where epithelial cells undergo a series of changes to acquire a mesenchymal cell phenotype. Now this may not have made any sense to you, but let me explain. So we have an epithelial cell, and as the name implies, you undergo EMT, and you become a mesenchymal cell. So what are some characteristics of an epithelial cell? Well, first off, they have high levels of a protein known as E-cadherin. Now, E-cadherin is simply the glue that holds these cells together. Think of it, there's a cell adhesion molecule. So these epithelial cells have high levels of this, so they're going to stay close together. While these mesenchymal cells don't. They have low levels of E-cadherin. So they're going to be distant, they're not going to have any colonies. So as I said, epithelial cells have high cell adhesion because of the high presence of E-cadherin, which is a cell adhesion molecule. Mesenchymal cells have no cell adhesion because of the lack of E-cadherin. Epithelial cells form cell colonies, so they're going to be bunched together. Mesenchymal cells do not form cell colonies, so they're going to be independent and individualized. Most importantly, epithelial cells are stationary. They're not going to move on their own. So, when scientists looked at this, they knew that these cancer cells were epithelial. But they also knew that epithelial cells were stationary. So how do they move? They move because they transition into a mesenchymal state, which is extremely migratory and likes to move. So EMT is known as the forward process from an epithelial cell to a mesenchymal cell, but it's also possible for MET to occur, or mesenchymal to epithelial transition. So the mesenchymal to epithelial transition is simply the reverse process of this. This will be significant later on in the presentation. So how does EMT play a role in metastasis? Well, first off, this is your standard normal epithelium. You have nice cell adhesion, you have nice cell colonies, and a nice organized cell structure. As you can see, you have your basement membrane, you have your normal epithelial cell, and then that red dot is the E-cadherin. It's gluing the cells together. But now, tumorigenesis is going to occur. So now, these regular normal epithelial cells are going to become epithelial cancer cells in your primary tumor. So this is the initial formation of a primary tumor, and cancer begins. After this, EMT is going to occur. So these epithelial cancer cells are going to become invasive and metastatic mesenchymal cells. Now, as I said before, this is just the normal metastatic process. The invasive and metastatic mesenchymal cells are going to invade the blood vessel, and they're going to invade a distant organ site. However, the main difference now is that once they reach a second organ site, they're going to undergo the mesenchymal epithelial transition. The mesenchymal epithelial transition, as I said before, is a reverse process. And you may be wondering, why do they need to undergo this? Well, let's look back at the properties. The epithelial cells are stationary; they proliferate very well, but they can't move very well. So they're not an ideal agent for metastasis. Mesenchymal cells, on the other hand, they move very well, but they can't stay put. They can't settle down. So once you reach a secondary organ site, we need to settle down again. So in order for these mesenchymal cells to settle down again and establish a secondary tumor, they must become epithelial cells again. So they revert back to their original epithelial state in the process mesenchymal epithelial transition. So how is EMT induced? Well, EMT transduction, this is a EMT transduction diagram. This may look very complicated, but it really is not. First of all, we start off with an extracellular signal. So here in the box, I've highlighted TGF-beta, which is simply a growth factor that is present in the tumor microenvironment. It's made by the surrounding tissues of the cancer cell. So this TGF-beta is going to act as a ligand to the receptor known as ALK5. So TGF-beta is going to bind to the receptor and activate an intracellular signaling pathway. In this case, it's going to be the SMAD protein signaling pathway. So once the signaling pathway is activated, it's going to activate intranuclear signaling. So I've highlighted in the box the snail, twist, and ZEB transcription factors. These transcription factors, they function by the targeting of E-cadherin, which, as I said before, is necessary to maintain the epithelial state. So once these transcription factors are activated, then E-cadherin is suppressed and EMT follows. So how do we target EMT in a pharmaceutical setting? Well, there are several possibilities. First of all, we can inhibit the EMT-inducing signals from the tumor microenvironment. So how do we do this? As I said before, we had TGF-beta. So if we block TGF-beta and we don't allow it to bind to the receptor, we can't allow, the intracellular signaling pathway will not be activated. So if this pathway is not activated, the transcription factor is not activated, then EMT does not happen. Now, we can also directly block the signal transduction pathways that relay to EMT initiating signals. So we allow the ligand to bind to the receptor, we allow the pathway to be activated, but then we inhibit certain points in this pathway to make sure that the EMT signals don't get from the intracellular matrix to the nucleus. Because as I stated before, the nucleus is really where the primary action occurs. It's where the transcription factors do take their, it's where the transcription factors activate and where they act and repress E-cadherin. Now we can also target cells while they're in the mesenchymal state. So we allow EMT to occur, but then while they're traveling through the bloodstream, we target and kill them. Now we can also block the mesenchymal epithelial transition to prevent colonization. So we allow EMT to occur, we allow mesenchymal cells to travel to a distant tumor site, but we prevent them from colonizing and establishing a secondary tumor, so now metastasis won't occur. Now, what is the impact of this EMT research? Well, we have revolutionized how we view cancer. Before, we had no idea how metastasis occurs, so we couldn't treat it. And that is why metastasis is currently 90% of cancer-related deaths. So now, understanding EMT, we finally understand how metastasis occurs and why metastasis occurs. So now we can develop specific treatments to target the metastatic process, so we can prevent the spread of the cancer before it even begins. Thank you for listening to my presentation. I hope you enjoyed it as much as I did.